Hello everyone, welcome to Pathomad. In this part 2 of blistering diseases, we'll cover bullous pemphigoid. Bullous pemphigoid, these are characterized by tense bullae. There are tense bullae since it is an subepidermal split as we have studied in the classification. So this above the split, there's an intact layer of epidermis, hence these blisters do not rupture. So they are tense bullae. The clinical image you can find the tense bullae containing clear fluid. Most common sites are flexor surfaces of the forearm, axilla and groin. It is an autoimmune disease, so this formation of autoantibodies, also IgG type, against the basement membrane proteins called hemidesmosomes. Here, for example, I draw this basement membrane. On this, I have this basal layer, cells of basement membrane, for example, and this is the basement membrane. Resting on this are the cells. These cells are attached. These basal cells are attached to the basement membrane with the help of this hemidesmosomes proteins. These are the hemidesmosomes proteins. These are the hemidesmosome proteins that bind the cells of the basal layer to the underlying basement membrane. So the hemidesmosomes have certain components. One such component is the bullous pemphigoid antigen. BPAG is one of the components of hemidesmosomes. Hemidesmosomes are required to bind the cells of stratum basale to the underlying basement membrane. When these hemidesmosomes are attacked by the autoantibodies, that is subsequently BPAG proteins are attacked, there is a split between the cells of the basal layer to the underlying basement membrane, and hence this is called a subepidermal split. And within this is a microscopic image showing you subepidermal split. All the layers of the epidermis are intact: corneum, granulosum, spinosum, and basal. Beneath this epidermis is a split, and this bullous cavity, it is filled with eosinophils. This orange pink cells, orangish pink cells are the eosinophils. When you go into the immunofluorescence studies, here you can see a subepidermal split, and also there is a linear Ig, that is immunoglobulin deposition, the linear pattern, the roof of the cavity. This is called a linear or ribbon candy appearance of IgG deposition towards the roof of the cavity, which is characteristic of bullous pemphigoid. So bullous pemphigoid, there is formation of tense bullae, IgG immunoglobulins, they attack the hemidesmosomes. Hemidesmosomes are present between the cells of stratum basale and the underlying basement membrane. Hemidesmosomes are made up of BPAG, and BPAG are attacked in this bullous pemphigoid, and hence there is a subepidermal split. In the subepidermal split, there are lots of eosinophils. In immunofluorescence studies, you find this linear or ribbon candy IgG deposition towards the roof of the cavity. These are the important points of bullous pemphigoid. Next, we will study about epidermolysis bullosa acquisita. As I have explained earlier, this is the basement membrane. Upon it are the cells of stratum basale. So the cells are attached to the basement membrane with the help of hemidesmosomes, and beneath the basement membrane, this is the dermis, the collagen fibers of the dermis. This is the dermis, and also the basement membrane attached to the dermis with the help of anchoring fibrils. Then these are the hemidesmosomes that attach the basal cells to the basement membrane, and the basement membrane itself is attached to the dermis with the help of anchoring fibrils. And these anchoring fibrils are made up of collagen 7. Type 7 collagen makes the anchoring fibrils. Anchoring fibrils attach the basement membrane of the epidermis, entire epidermis, to the dermis. In this way, the epidermis attaches to the dermis with the help of anchoring fibrils. So, in epidermolysis bullosa, there is a defect in this gene called COL7A1 gene, which encodes this type 7 collagen. Type 7 collagen needed for anchoring fibrils. And hence, when there is a defect in type 7 collagen, the anchoring fibril is defective. So, the adhesion between the epidermal basement membrane and the underlying dermis is lost. So, there is a subepidermal split here. See here, this is the basement membrane. Resting on it are the basal cell layers. The cells are attached to the basement membrane with the help of hemidesmosomes. The entire epidermis is attached to the underlying dermis with the help of anchoring fibrils. This is the microscopic image showing you the subepidermal split. This is the entire epidermis, stratum corneum, granulosum, spinosum, and basal. Beneath the epidermis is a split. Between the epidermis and dermis, there is a split, subepidermal split. And into the immunofluorescence studies, there are linear deposits in the floor. There's a floor. This is a bullous cavity. In the floor of the bullous cavity, there's a deposition of immunoglobulins. Coming to dermatitis herpetiformis, the most common sites involved are the extensor surfaces of the elbow, knees, buttocks, and upper back. The elbows are involved. These are symmetrically involved and bilateral involvement is seen. The most common association is with the celiac disease. So the association celiac disease gives you a clue towards the diagnosis of dermatitis herpetiformis. Since it is associated with celiac disease, there is IgA involvement, IgA deposition in the subepidermis. When you go into the microscopic view, there is deposition of fibrin and neutrophils at the tips of dermal papillae, forming a papillary microabscess. This is the entire dermis. This is the entire epidermis. This is the dermis. So this ridge, this protrusion of dermis into the epidermis is called papilla, dermal papilla. See, this entire thing is the epidermis. Epidermis also projects into dermis. This projection is called rete ridges of the epidermis. Similarly, the dermis also makes projections or protrusions into the epidermis. This is the dermis. This protrusion of dermis into the epidermis, this is called a dermal papilla. Dermal papilla. So here we see lesions, that is presence of neutrophils, fibrin depositions in the tips of dermal papilla, forming papillary microabscesses. Here this is the tip of the dermal papilla. You can find the inflammatory infiltrate neutrophils here. And also there is focal separation. You can find a focal separation between the dermis and epidermis, focal dermo-epidermal separation, which ultimately all this coalesce to form a subepidermal blister. In the immunofluorescence, you see granular IgA deposition. This is a granular IgA deposition. The tips, this is the tip of a dermal papilla. In the tips of dermal papilla, you find deposition, localized granular deposition of IgA. Let us summarize all the blistering diseases once again. Firstly, in pemphigus vulgaris, there is involvement of desmoglein 1 and 3. 1 and 3 are involved, and the split is suprabasal split, that is a row of tombstones. And in foliaceous, there is only one involvement, desmoglein 1, and subcorneal split is seen. And both these pemphigus groups, they show fishnet pattern on immunofluorescence. In bullous pemphigoid, BPAG, which is a component of hemidesmosomes, these are attacked and they show you ribbon candy appearance on the roof of the cavity on immunofluorescence. In epidermolysis bullosa acquisita, there is defect in collagen 7, collagen type 7 collagen is involved, which is the component of anchoring fibrils. In both this, there is a subepidermal split. Both of these show subepidermal split. And the immunofluorescence, the linear immunofluorescence deposition on the floor of the cavity. In dermatitis herpetiformis, IgA is involved, associated with celiac disease. Also, it shows a subepidermal split, and there are tip lesions, IgA tip lesions, tips of the dermal papillae. This is to summarize all the blistering diseases. Thank you, everyone.